Okay. I mean, if you can hear me, as always, I leave the chat, then I can see. Okay, so it's working. It's good. Okay, so lots of people waving their hands. Where are we? Going to the item analysis. This is the review of our exam. Okay. Google prompts here. Click here. Okay, so let's see what we have on our test. So the first of all, I mean, most students done well during the during your exam. I'm pretty sure some people very happy with the result, and some people are not. I mean, always the exam give that motion. Okay, so this is the this is your first exam. It was last March. At that time the median scores was around 10, so it was half about the score. And the second exam, I can see some students have higher score than before. So quite lots of students get like 12 or 13. So I can see the mid range around here. And also quite some people have more than 15, so which is quite encouraging. And if you look at the exam scores, the first one and the second one, then if I get the diagonal line, then I can see upper part, which is the improvement of your score. So I mean, I'll say that's quite positive. But of course, some students get lower score than before. So students on these parts, you should probably be more engaged in the activities, then you can check your understanding. So I mean, that would be my best advice for your learning exercise. Okay, so that's good. And as you know that the second exam will be the 20% of your total score. So and the third exam will be the same, and the last one will be the 30%. So keep in mind your proportion, then you can schedule your exam scores. Then we have the assignment. I think due is today, so please submit yours on time. And you probably see the formats of your assignments. And I can see several students already submit their assignment, and I was quite surprised because given the the last years or the any previous years, I mean this year I only look at the 3 students submission, but I mean absolutely fantastic. So I'll see more sort of better product from your writing. So that's what I'm waiting for today. And then another things I'm asking you is the is the survey. So I did lots of requirement for you. So if you click here, and then this will give you the survey for the mid-class. This is asking you whether the tracking your things properly or not. And the university, they gave you the mid-semester survey through the Blackboard, but the Blackboard is amazing system, so you cannot use Timely. So only I think 5 or 6 students on the survey. So I mean, it's unlikely improve my course. So I mean, it's a bit sorry for asking you again, but if you can do sort of mid-semester survey or mid-class survey sometime this week, and that will be really appreciate in helping my class and also organizing your learning schedules as well. So I'm sort of develop some questions about your classes. So hopefully that works for you. Okay, and then the exam review. So the second exam, and I I mean, I can see quite, you know, most of the students, they get really comfortable with the questions at this time compared to the first exam. And also, I say quite many questions are quite, you know, straightforward for you're taking. So that's good. And then you probably get the, you know, the positive scores from the some core questions. So, I mean, I would say there is another encouraging thing. So, because the main purpose of the third session was about understanding the population structure and how mutation working in that, you know, the shaping populational differences. And I would say quite most students get the sense. So that is good. And of course, always some questions are quite tricky. So for instance, like this question. So this is about the LD situation in our the chromosome 6. And that was good. And some of the choices quite easy. So for instance, like, you know, you can use some text SNPs representing the, you know, the major 3 haplotype blocks. And also you can use the text SNP for microarray, but also you can use for the whole genome sequencing. But we are not using this because of the expense, right? So I mean, direct sequencing is always expensive. So we can choose more efficient way to do that, right? And also LD is sort of the continuous information, so you can put more sub LD structure within the large LD structure, right? So that is the asking that issue. And microarray, this is sort of the same, you know, choice as this one. So that's another the same value. And also MHC region kind of represent population differences. This is basically asking you whether you understand LD is representing the population differences or not, right? So MHC region doesn't really matter in this context, right? So there was, I think that is most tricky things in your exam. Okay, so back to the question. And another interesting and also tricky question was this one, right? So I think most students get the good answers, but like, you know, de novo mutation alters 2 copies of the gene, right? De novo mutation always work in a heterozygous format. So that is one thing, because the, you know, think about the strands of the mutation, because we got the 2 strands from each parent, father and mother, right? And the de novo mutation arising from one of the parents, not both parents, right? But there are unlikely that things happen in our biology, right? So de novo mutation happens at the 1 strand of the parents, and that is the format of the heterozygous, right? So that is asking you that issue. And other than that, it's all, I mean, quite vanilla. So that is good. And another tricky things, okay, so sort out by difficulty. So I think these 4 questions probably most tricky things. And other than that, the difficulties is quite rock it out. So it's quite simple. Okay, so back to the Marfan syndrome question. So that is the question I tweaked from the first exam, right? So updates few of options for that, right? So this is asking you the aspect of the population and mutation and additive and dominant situation, right? So okay, so in the family of both father and son, diagnosed expected, yeah, this is the asking the segregation probability always 50%, that's cool. And you can calculate the genetic variance of dominance factors using father and children with diagnosis, right? And that is the genetic variance, right? So using the additive factors, and if you have sufficient numbers of families, that is basically asking you heritability, right? And if the Marfan syndrome follows autosomal dominant inheritance, right? And this is asking you whether dominance and the frequency not related to each other. This is the basic questions from the Yu's lecture, right? Punet's lecture and Yu's question. And if mutation does not alter fecundity, then the mutation could be passed to the next generation. This is asking you about the fitness, right? Okay, that's fine. And then it's the same question. Okay, so that was the things quite some students missed the opportunity. So coding mutation occur in an exon, so that's for sure, right? And the coding mutation could be classified as non-coding mutation by different transcripts. So this is the asking you whether you understand the plan or the scheme of the variant annotation, and that is based on the multiple transcript, right? So that is yes. And the new non-coding mutation would not impact on coding gene transcription. That is wrong, because the in non-coding genome, we have the regulatory elements like enhancer or promoters, and that's definitely impacting on the gene transcription, right? So that is the incorrect thing. And coding mutation generally show larger phenotypic consequence than non-coding variants. We saw that example in the slide. And missense mutation of gene A and synonymous mutation of gene A should have the same length of protein A. So this is basically asking you whether you understand, missense mutation alter one amino acid sequence compared to the nonsense mutation or stop codon mutation, right? So that's basically asking you about the protein consequence. Okay, so I think these 4 questions are probably most tricky thing. And if you have, if you have some questions still not clear after you review the answer sheet, it's always welcome to leave your questions on the Slack channel. Then you can follow up that. And also, the exam 3 and exam 4 also related to the session 2 and 3. So if you're not clear about some stuffs on the session 2 and 3, it's always encourage you to leave some questions and comments on the Slack. Then you can understand the topics on session 4 and 5, right? Okay, so that's all good. And back to the our lecture slide. Okay, go away. Okay, so we are moving on to the session 4 and 1. Before we doing this, we can look at what we are going to learn in session 4. Okay, so in the session 4, this is about the human genome. This is my expertise and also the topics I'm most excited. And we are covering 3 topics from this session. So first one is about genome, and the second one is about the next generation sequencing. This is the latest genomic technology in the field. And session 3 and 4 is about the data format of the next generation sequencing. So modality means that format, right? So data modality means that, if you want to study in genomics, then you have to understand what kinds of data set you can deal with in your career, right? So what I find most difficult things when I started learning the genomic technology was, to understand the format and the types of the data set in the genomic field, right? So from the session 4, 3, and 4, we are covering the basic, data files and data, types of the NGS format, and including like, whatever FASTA-Q or SAM or VSET format, and that is the mainly on the session 4 and 3 and 4, right? So this session 4 is all about the human genome, and then after you are learning this session, then you are quite comfortable with dealing with the latest genomic data set, right? And then you are ready for handling the larger scale data set with the programming language, right? Okay, so this is sort of the entry session for that. So the first topic is the genome, right? So what we're going to learn today is the 4 topics. So first one is of course, about the genome, right? So basic concepts of that. And the second one is the differences between the coding and non-coding genome, right? And the third one is we are learning the overview of the Human Genome Project, which is the major milestones in biology science, right? And the fourth one is the initial impact of the sequencing of the human genome in our biology, right? Okay, so first disclaimer is that the majority of the slides actually borrowed from the lectures by Professor Aaron Quinlan in Utah. I personally know him, and he's one of the great bioinformatics researcher in the world. And he's, another good thing is he opened all of his lectures on GitHub, as I do. So I borrow some of the slides from him, and he's okay with that. And also, if you are interested in sort of applied computational genomics, you can go to his slide and look at some extra materials for your learning. That's always recommended. Okay, so first one is the genome. So we're going to learn about the basic concept of the genome and the differences between prokaryotic and eukaryotic genome, and talking about the euchromatin and heterochromatin, right? So genome, we normally refer a complete set of the DNA in an organism, right? So all of the living organisms, we have the genome in our body. So genome contains genetic material of an organism. So some people are asking that, the genome is the sort of the sets of some instruction, how body and how activities is going on in our living process, right? And genome contains DNA, but also genome also consists of RNA, like COVID-19 virus, right? And genome is the system that contains the instruction needed for human development, and also it's directing the activities of nearly all living organisms, right? So that means that if you're looking at the genomes, you typically think about the genome is like linear format, and it's wrapped up as a the chromosome structure, and this sort of the linear information of the A to Z C contents in your genome. This is sort of the typical things, but in a living situation, genome is actually dynamically working, right? So if you're thinking something, or if you're talking something, or if you're moving something, your genome is always working. So it's dynamically wrap up or wrap down the things and transcribe the new protein, and makes the new protein and working on a specific activities, right? So now I'm talking to you about delivering some lectures, and in my brain the genome is working, and it gives you some of the gene transcription to think about, or to use the memory to remind some materials to give you as a speech, right? All of this process always operated by the genomic consequences in our genome, right? So genomics is the study of the genome. So if you study the patterns, or if you study the basic characteristics of DNAs, or if you study the dynamic patterns of the DNA or RNA, that's all the sort of disciplines we call genomics, right? So I would say genomics is one of the leading the field we are facing in modern biology, because we well know about the central dogma, we well know about the process from the DNA to protein to phenotype. So people want to look at the foundation of all of this process, and that's always pointing out by the applications from the genomics, right? Okay, so if you look at the prokaryotic genomics, and this is one of the simple and single-celled organisms, and we know that it's lacking their nucleus and also membrane-bound organelles, right? And it's quite simple operations to do their living, right? And the majority of prokaryotic DNA is found in the central region of the cell and called nuclei. And it is typically consists of single large loop called circular chromosome, right? And also you're probably quite familiar with other, the studies like microbiology, right? And most of the DNA sequencing code for protein or RNA, and I mean, it's often known as a lack of introns, small amount of non-coding DNAs, and also that means the regulatory sequence is rather simple. So at this point, people discuss about why eukaryotic genomes, or why humans or primate evolutionary higher organisms, they have most complicated format of the regulatory sequence because they're doing something more complicated activities than prokaryotic organisms, right? So eukaryotic genomes is always to the contradictory, like prokaryotic cells, eukaryotic cells have a membrane-bound nucleus, central cavity surrounded by membrane that house the cell genetic materials. I mean, this is quite obvious reason because you have to keep it in the safe place, then you can do more stable activities, right? So if your genetic materials posed in the situation like easily exposed to the external situation, then your information unlikely be stable in lots of different activities and also different environment. So evolutionary, the higher organisms, they always put their the important materials in a secure place, that is the membrane, right? And the number of membrane-bound organelles, and there is compartments with a specialized function that flow in the cytosol. And organelles means little organ, and thin name reflects the organs, like the organ of the body, have unique function as the part of the larger system, right? So, I mean, you probably learned from the symbiosis situation in a cell's evolution, right? And it has the multiple linear chromosomes because over the long evolutionary process, we are separated into the different chromosomes and put it into more discrete functional systems, right? I mean, the class is not about the cellular biology, so I'm just reading whatever I put the boring overview. So eukaryotic cells are much more complicated than those of prokaryotic, I mean, of course, and they are packed with fascinating array of cellular substructure that plays important role in energy balance, metabolism, and gene expression. And always, 101 class image for the animal cells and plant cells, we are memorizing all of these stuff, but you're not gonna use this information in your career. It's always in the Wikipedia. But surprising things, what I found recently, if you use the super resolution microscope, and if you look at the the cells, cells obviously all of different formats in a different context, right? And mitochondria and ribosomes is quite different what we usually see from the textbook, right? The mitochondria genomes are quite linear. I mean, the most of the mitochondrions we see in the cells are like tiny little worms or a bit of rounded things, right? But this is actually like very linear things, and it's like very much like a layer structure. And that was quite surprising things to me, because we never thought about it. We're always looking the textbook image, but real world examples from the latest technology gives you all different things, right? So I mean, you can understand, we have so many compartments in our cells, and genomes is one of the things. But other than that, we probably more further detail from the cellular biology stuff. Okay, so how many genomes exist in a human cell? We have one nuclear genome in a human cell. So I mean, that means we have multiple nuclear genomes in multiple cells, right? So, I mean, people are talking about, typical human is comprised of roughly 40 trillion human cells. And I mean, people talking about, our DNA is 3 billion base pair, that's for sure. And if you stretch out each haplotype genome would be roughly 2 meters. And people always, give you, classic examples or joke about, if you stretch out all of your DNA from your cells, they wrap up, 20 times the globe at the Earth. But I mean, I would say if you stretch out your DNA, you're gonna die, right? So, I don't know. I mean, whether that is the really fun things or not. But anyway, so we have really long linear structure of our DNA in our cells, and that is dynamically wrap out or wrap up during some certain task, and that has sort of certain purpose as well. But we also have few exceptions of this linear structure. That is, one classic example is the red blood cells. They are lacking out the chromosomes. And also, if you look at the cells in our liver is somewhere in central part of our body, right? And for your alcohol or whatever you're smoking or the drugs, and that's always, the wipe out in your liver. And so liver has to be much more fragile to that, not fragile, it has to be much more plastic to that situation. So in that case, liver cells are based on the polyploid. So it's more than haploid genome, and that is easily adapted to that situations, right? So liver cells is one of that issues as well. And also we have many mitochondria genomes. So we have numerous mitochondria genomes in our single cell, right? And also these mitochondria genomes are different composition across different tissues, right? If you look at livers, if you look at kidneys or brains, they have different numbers of mitochondria because we have different requirements of energy during the biological process, right? So if you want to generate more neuronal cells, which is the brain cells, then you are requiring more ATP energies, right? So that case, neurons use more mitochondria proportion than others. That case, you have more mitochondria genomes in that cells, but some of the cells, they don't really need many energies on that. So that case, way less number of mitochondria genomes in their cellular composition. Okay, so if you look at the genomes in the context of the cells, another things you have to consider is the euchromatin and heterochromatin, right? So to think about the situation when people look at the genomes in the cell at the first time, that is probably because of the development of the microscopy and the staining technology, right? So if you stain your DNA or the genomic material, then you can see their existence by your eyes, right? So in that case, you have to aid by microscopy technology, right? So in that case, you have to stain, you have to color out some of the part of the genome. In that case, we can separate into the two parts. So first one is the heterochromatin, and the second is the euchromatin, right? So whenever I explain these stuff, I always remind that which one is open and which one is closed, right? So the memorizing things is always not working. So that means the heterochromatin is the most abundant cells that are less activate or not activate. So that means the closed format, right? So it's the keep it in the secure format. So unless you get some signals to open up and to do something, then the genetic material is actually wrapped up, right? So that is the stay as a closed format. Then if your cells get the signal to do something, right? So because the genetic material is the result of the environmental exposure, right? So the situation like if you want to think about something, then your brain cells get the signals from somewhere, and the genes or the proteins related to your cognitive function has to be transcribed that case, the part of the genome containing that genes or protein molecules that has to be opened up, right? So that situation we call the euchromatin, right? So euchromatin is the prevalent in a cell that are activate and in the transcription of their genes, right? So for instance, like, as I explained that, if you think something, if you want to say something, then your brain cells get the signals from from somewhere, probably from the another brain regions, then the cells has to be activated and the genes related to that biological process is open up and then do the gene transcription and make some protein to do that, right? So it's always, having a interesting topic like, how long memory is stored in our brain, right? So if you, sometimes you probably think about certain situations like, if you have really, good moments in some day, right? Sometimes you memorize that situation, you memorize that, memorize the taste, you memorize the smells, or memorize the, the weather, or memorize the place, and that memory has to be stored somewhere in your brain, right? So we want to know about where it has to be stored, right? So you always think about, probably that's somewhere, some brain regions in our brain, right? Like, hippocampus, right? Hippocampus, and then some layers of that, hippocampus toward that memory, right? But interesting stuff is that, it's memory, it's always different format, right? So it could be sometimes like, taste, sometimes like, fragrance, or sometimes like, some visual input, right? But how that can, stored as a, certain format in certain place, right? For instance, like a hippocampus, right? So how that memory things stored in the hippocampus. So one, hypothesis is that, those memory things is stored as a, protein, or as a, epigenetic things, or as a, temporarily RNA stuffs or something, right? So all of these, biological molecules can be stored in the situation. So in anyhow, like, RNAs or proteins has to be expressed, has to be transcribed, or has to be translated to store that memory things into your brain, right? So all of these things, all of that activities has to be happen in a active cells, which are, we can call euchromatin, right? So that case, we can say that, the memory related activities, and that is the linking to the euchromatin situation. But if you see the differences between the heterochromatin and euchromatin, the first things people want to know about is that how much the genomic materials is the active in the cells or inactive in the cells, right? So that case, people always looking at some classic examples from some biology, right? Okay, open link. Oops, it's gone. Ah, that's great. Okay, so it's gone. Sorry for that. I think it should be working, but it's not. I hope Yale University is not gone. Hopefully, it's gone. Oh, or the university website is probably gone. Oh, is that working? Yeah. Google is working, but Yale University is not working. I I hope the link is working somewhere in the working hours in Eastern time. Yeah, hopefully. So that time, I'll try in maybe 6 or 7 hours later. Okay, so then in the meantime, we move on to the next topic. So next one is the coding and non-coding genome. So this is the differences between which genomic regions give you the protein or protein coding genes, and the other one is not, right? So if you look at our genome, some of the regions are coded for the proteins, right? So the genome contains DNA sequence for amino acid and protein, we call the coding region, right? And it takes about 1% of our genome, right? So, I mean, that could be a surprising number for you or not, because, you know, I mean, think about the entire genome, only 1% of our genome makes you the protein, right? So protein is always the sort of the unit of some phenotypic development, right? So think about that situation, only 1% of our genome doing that. So some people think that that's quite inefficient way, or some people say that that's quite, you know, the mystery things for the evolution, right? So, but anyway, so looking at the coding genome, we have some, you know, the rules to make some protein sequence, right? So we are based on the triplet codon, right? So triplet codon is the 3 base pair, the units to make a 1 amino acid. And what, some of these triplet codon that encodes the starting position that we call the start codon, right? So that is the obviously the first codon of the mRNA transcript, and that is the ultimately translated by ribosomes, right? And the stop codon is a nucleotide triplet within the mRNA, and the signals terminates of the translation into proteins, right? So we're not going to get the endless length of the protein in the translation. So it has to have the signals for the stop, right? So that is the stop codon. And predict a coding sequence by finding start and stop codon. So at the beginning of the genomic research, what people normally do was looking at, you know, which area is the start codon and which area is the stop codon. Then people easily track down, okay, probably from this and this, I mean, these regions probably the protein encoding area, right? So people easily estimate the coding sequence. And I mean, these things is not really surprising things anymore, because we now have the reference genomic sequence for the human genome, but at the very early times, like, you know, 80s or 90s, I mean, people doing some research on finding, you know, or estimating the start and stop codon is something, right? But this is not surprising anymore. Okay, so if you look at the coding regions, you know, genome, we can further details, like, you know, coding sequence, or shortly we call the CDS, or we have the exon, we have the intron. So exon is the actual regions can have the protein codons, right? And introns is the area between the 2 exons, right? So, and then the existence of introns, you probably learned from the molecular biology. Introns always give you the diversity of the evolution and also the transcription situation. Then you can give you much, you know, diverse option for your gene transcription. Then the organism can easily adapt it to certain situation, or can have the complex the process in a development as well. Okay, then the transcript is a sequence transcribed from the DNA into the mRNA, right? And they can have multiple different transcripts because of the alternative splicing. Okay, so we looked at the coding and non-coding, coding sequences at the last time, and we can look at back about the transcript, right? So if you go to this link, this is called the GenCode. So we have different database to show how many transcript and how many genes in our body, right? In our genome, right? And I mean, different database means that, you know, across the database, we have the different information about number of genes and number of, you know, transcript and the names of the genes as well. This is like situation, you know, I mean, which units we are using for a certain situation. So for the gene annotation, we have the same situations as well, because, you know, some European countries you use this database, or some, you know, American database use this information. Of course, people try to unify this different database, but still, you know, I mean, if you look at some studies, they use their own preference over some database as well. So I normally use the GenCode because GenCode is most comprehensive database, which means that it has the largest number of transcript and protein coding genes. So I normally use GenCode. So I give you the examples of the GenCode. So if you look at the stats about the current human genomics, then you can see how many genes in our genome, right? So total number of genes, we have 60,000 genes, right? So I mean, quite large number. And we have protein coding genes, we have about 20,000 genes, so less than 20,000 genes, right? And knowing the number of genes is, one of these scholastic discussions at some point. So the people in 90s, people always discuss about, do you know how many genes in our genome? And that was one of the most hipster things in their discussion, but this is not new things anymore. But anyway, so if you look at other types of genomes, like non-coding RNA genomes, small non-coding RNA genes, and some pseudo genes, so like nothing in there, and some genes like for working in an immune system, like to fight with the COVID-19 or something. So this, all of the numbers, we kept it in here. Then if you look at the transcript, we have much larger number than just the number of genes, right? So we have 240,000 transcript. So this is about 4 times higher than just the number of genes, right? So you can also look at the details of that. Okay, so that's all. Then if you look at SN2A examples, then if you go to here, and this is the one of the European database as well, and they're called ENCODE. And if you look at the gene name SN2A is one of our friend. And if you look at this, then you can see the transcript table. And this is the same gene, but it has 1, 2, 3, 4, 5, 6, 7 transcripts, right? And out of these 7 transcripts, 1, 2, 3, 4, 5 transcripts coding proteins, right? So these 5 transcripts can do protein coding translation, right? But these 2 transcripts doesn't do anything. These 2 transcripts doing nothing, just transcribed as a retained intron transcript, right? So that is not subject to translation. It's just an mRNA and disappeared after transcription. So if you look at the transcript, then you can clearly see the differences, right? So obviously the transcript name 201, oh, it's really hard to read. So 201, and that is the longest one, right? And the second one has the almost same, but probably different somewhere in here. My eyes is not working. Yeah, somewhere in here. Or probably one of these exons they swap their position. So that's 1 and 2 has differences. And then the third one has the shorter form, right? We don't know why it's shorter, but it's short. And it is probably doing some different functions in a development, right? And then, okay, so this is another form of the first one, right? It used to be called like this, right? And the second one also used to be called like this. And the fifth one is the retain intron. So it doesn't have the start and stop codon. So it's not gonna process to the translation. And the fourth one is the intron, retain introns as well. So you can clearly see the different formats of the transcripts even in the same gene. And these things is always happening in other genes as well. And if the gene has much more diverse role in a development, in that case, they are likely to have more transcripts than others, right? Because they has to be versatile. There has to be diverse role in a development, right? So let's say gene A has lots of talents, right? So like, human, so it can work in a kidney, it can work in a liver, it can work in a heart, and they're likely to have more transcript than just one, right? Because each different transcript has to work in a different situation. But if the gene working in a one situation, for instance, like, gene B, and that is only working in testis, then in that case, the gene likely have only one transcript, because there is no point it has more, more than two transcripts, right? Okay, so there is the examples of the different transcripts. So moving on to the non-coding region in our genome. And non-coding genome is literally means that do not code for proteins, right? And it's, again, surprising things or not, it takes 99% of our human genome. So if you look at our genome, most of our genome doesn't code proteins. It doesn't do anything for the protein coding. That's the very early thought in a genomics. But nowadays, we now know that lots of the regulatory sequences exist in a non-coding genome, and that is working for gene transcription, right? So if you look at the gene like in here, so this is the coding sequence, right? But some of the regions like in here and here, that is the non-coding region, but that region contains some regulatory elements. That case, it regulates or it modulates the gene transcription of this gene, right? So how that can happen? The reason is that the gene transcription, as I mentioned that, the gene has to be transcribed in a certain situation. So for instance, like, if I'm thinking something now, then my brain cells has to express some proteins to do my cognitive functions, right? So for that situation, you have to transcribe your genes immediately and also work in a very transient situation, right? So how we can do that? There is no such person you can order your brain cells, let's do the gene transcription. There's no such things, right? So our evolution always takes the, the complexity development, right? So during our development, our, cognitive circuits, developed for that complexity and certain situation without knowing that, without, noticing that situation, our brains, automatically express some genes to do some functions, right? So that case, you have to transcribe your genes more efficient ways, right? So that case, you always get the help from the regulatory elements, like, promoters or enhancers. So if you think about the meaning of promoters or enhancers, that gives you to some doing some enhancing your functions, right? So that case, if you do in a gene transcription situation, these promoters or enhancers are diversely regulate the situation and do makes you to do something, right? So that case, higher evolution organisms like humans or like even primates, they have much more proportions of regulatory elements in our genome. So if you look at our genomes, we take lots of non-coding regions in our body, right? So how that works in our situation. So if you look at the linear structure of our DNA, so this is just like a linear sequence of the DNA, right? But in a transcription situation, it's forming out the tertiary structure. Tertiary means that the 3D structure. So if you want to transcribe the gene in here, then you have the non-coding regulatory elements called promoter, and also the enhancers in this part, right? So if you think about like in here, it's wrapped out like this, right? And then the promoter and enhancers get the proteins called transcription factors, right? So if you think about, the things you learned from the molecular biology, when the gene transcription acts, then RNA polymerase II binds on the promoter, right? and it initiates the transcription, right? But also the transcription situation is much detailed than just the initiation. It has the initiation, whatever the store or the back troops or something. But anyway, it's evolutionarily complicated, organized. And then this promoter and RNA polymerase II is helped out by some proteins called transcription factors. And these factors can modulate more finely in a certain situation. So if you want to express out some proteins related to your emotion, then some 2 or 3 transcription factors binding on the promoters. But some situation like if you want to express out the proteins related to your kidney functions, then different types of transcription factors bind out to RNA polymerase II. So in a different situation, different transcription factors makes a combination to achieve certain biological activities in each context, right? So in kidney development, liver development, or alcohol assumptions, or the movement, locomotions, or different situations, right? So all of different situations is like the different combinations of transcription factors and do the gene transcription, right? So that is the importance of the non-coding regions. Okay, so we are moving on to how people defining our genomic sequence from the large-scale milestone project called Human Genome Project. So we have the long history of genetics from Mendel to nowadays, right? So this is the figure of how genomics evolved from the Mendelian days to Human Genome Project, and long history and Mendel's and central dogma, and this is the one gene and one protein concept, and one hypothesis and one gene and one protein hypothesis, right? Bidder and theta, and blah blah blah, and the central dogma and the recombination vectors, right? Enzymes, recombination enzymes, right? And the first, the genomic technology, Senya sequencing, and beta Senya sequencing, and Human Genome Project, right? And this is the timeline for that. Okay. Okay, so we are covering this area, so Human Genome Project. Okay, so what I now I remember why I put this slide. So this is fun thing. So Human Genome Project is obviously the largest project we have. We means the human in our history, right? And how this Human Genome Project started, right? So of course there are some the regions to do this. So for instance, like technology developed, and technology was in the back end, so people can think about we probably can sequence human genome by our hands, right? So the obviously PCR developed when I born after a year, and then the Senya sequencing technology better and better improvement. And then, the money came in and some companies came out and they can do some sequencing. And that is sort of the technical, and that is the thing you probably heard from some the textbook. But another reason is that, in 1980s, late 1980s and early 90s, what happened in our world? The things was ended called Cold War. Cold War ended at the time. So Cold War is the war between the Soviet at the time, nowadays Russia, and the US was long history of the battle about whatever their power, and they showing off their power, and they technology developed for killing each other, right? And obviously the nuclear weapons or the all physical advancement comes from that the Cold War situation. And suddenly 1989 Cold War ended at the time, and we have no reason to more invest on that development, like weapons or whatever the high tech the algorithm things to the decode other military informations or something. So lots of physicists felt quite uncomfortable at the situation, and lots of physicists relocated into the different project that is called Human Genome Project. This is not conspiracy things from my head. This is actually from the scientist lecture. His name is John Quackenbush. He is one of the scientists involved in Human Genome Project. He is obviously the physicist, and lots of the talented physicists, they came in Human Genome Project, and they developed the algorithms for the genetic materials and also building the reference genome. The reason is that at the time we can only we can only do small scales of the PCRs and the segment sequencing. So that is very tiny fragments of the DNA information compared to 3 billion human genome, right? So people wants to predict our the entire human genome sequence out of this limited information. So what people do at the time, develop some algorithms like hidden Markov models or something that is the sort of the advanced algorithm development from the physics research. And lots of these physicists, they are engaged in algorithm development and also working in a largely different biological problems, and they ended up in human genome, the reference buildup, right? At the time, if you think about the 80s and 90s, as I mentioned, that at the time we can only do tiny fragments of the DNA sequence from certain gene. So this is quite tiny information compared to the information we get nowadays. The reason is, these days we can sequence one person's genome in a day, but at the time, people only can do 20 base pair or 100 base pair, only 100 DNA alphabets from the one assay. So at the time, people find some gene sequence. So let's say if you find the sequence of SN2A, that is your big party day, right? So if you find one sequence, then you can become professor, right? So because at the time you find one gene sequence that is really matter. So what people do at the time, they developing the database, they developing the curation, right? So if you find some sequence from some gene, so let's say you find the gene and you are working in a laboratory in France, then you report the sequence and you report to the GenBank database, right? Or if you find some gene and if you're working in Brazil, then you can also report your sequence and store in a database. So, 90s, 80s, and 90s at the time we are doing that, we did that, right? So at the time we stored the DNA sequence information in a GenBank and put it into that. Or some people bought the books about the gene sequence, right? So at the time, internet was quite bad and we didn't use computerware. So we just bought the books, having the DNA sequence in a genome, right? So, we couldn't really think about that because, these days you can go to the link in here, then you click 1 or 2 times, you can easily see their sequence, their positions or whatever their shapes. But in 80s and 90s, we kind of do that, right? And our computation is limited. So at the time, they sell the DNA sequence in a book or in a CD, right? So I just want to show you the time is quite, the flying and nowadays many things are different, right? So, but anyway, I don't know why I quite, excited about the, introducing that time, but at the time, that is probably my first computer, right? So we call the Sejongdaewang, and that is the, I think I got the first computer at the 1992 2 or 3, and there was like, 400, I don't know, Korean one is always bloody difficult. It's about the 400 million ones or something, yeah. And there was the Seijin company, and they always use the Jindo dog. There was the commercial about that. But anyway, this is not important things. Okay, so back to the Human Genome Project. So Human Genome Project, okay, so this is like, Hansarang genomic research at the time, but you're not going to have that one anymore in these days, so forget about that. So back to the Human Genome Project. So Human Genome Project is finally done in 2001, and that is done by 2 institutes. So first one is the public sector, which is by government, right? So that is done by the United States government, called the International Human Genome Project, HGGP. And then also meantime, private company did that the same things by their money, and they called the cellular genomics. And there is another name is the Craig Venter Institute. That is another private company based in the San Diego. And our one of our professors in our department, Stanley Kim, used to work in that company, right? Okay, so he is the living examples for that. But anyway, so International Human Project on a day, February 15, 2001, Nature published the paper about the Human Genome Project, and the same week, the library of the journal and Science, I think this is a better journal than Nature. The reason is I published there. So anyway, Science published the paper from the company, the private company, Cellular Genomics, and they also reporting a draft human sequence based on their own data set, right? And as I mentioned, that this is the draft sequence, so it has to be the first version. It has to be under improvements. But anyway, this is the sort of first sequence we can use as a reference for the human genome. So we normally call it the reference genome. So what reference genome does? So reference genome is the sort of the complete sequence of the human haploid genome. So in the previous situation, if you think about the previous situation, we only have the snippets, which means that we only have the fragmented information. We have very tiny part of the DNA sequence from our genome, right? So this time we have the complete set, right? So if you play game, then you could play only 1 or 2 chapters as a trial. So in the old days we are in that situation, but now we have full addition for that. So now we have all of the sequence for the the DNA sequence and location of the protein coding genes. So if you think about 1913, like 100 years ago, if you think about the Morgan, and they look at the genes on the X chromosomes, but they didn't know anything about the genomic locations and their positions other than linear fashions of the X chromosome. So this time, if you have the reference genome, now you have all of the genomic position from the human genome, right? And reference genome is working as a map, right? So if you think about the map, if you travel somewhere, right? So if you have the map, then you can easily track down, okay, so this restaurant located in here, how far they are, how can we get to there, right? But if you don't have the map, you're not going to have any idea about the locations and positions and the existence of the elements in the place, right? So reference genome is working in that situation. So if you know about the the position, if you know about how many proteins in our genome, then you can easily think about the genetic architecture of the human disease, and also think about how human genomes evolved over the time as well. So reference genome giving you the information about we can think about the human genetic research in the field. Okay. Oh, this is the same slide. Okay, going to here. So reference genome doing some great job in this situation. So nowadays we can, I mean, as I mentioned, that we do the sequence of the single individuals in a day. So if you go to clinic, then you can ask, I want you to get some, DNA sequencing. Then you get the DNA sequence information. Then once you get the DNA sequence information, then use the reference genome and compare to reference genome sequence, then you see which part of your DNA sequence has the mutation differences or some deletions or insertions. All of this information can be made from the reference genome, right? Because we know that sequence, then we can easily track down individual's genome, right? And of course, not just the individual genome. If you look at the multiple people, let's say population, then you can also easily track down, how many people having their insertion, how many people having their SNPs, how many people having their deletion. Then you can easily compute the frequency of the allele in a genome, right? Okay, so let's think about the technology for Human Genome Project. At the time, people used the technology called the Senya sequencing. So for the Human Genome Project, we don't have the latest technology called next generation sequencing. So at the first time, people used the Senya sequencing. So this is the technology sequencing by synthesis, not by degradation. So basically, you use the radioactive primers hybridized to DNA. So you mix the DNA samples with the radioactive primers and polymerize and nucleotides with the terminator. Then you put it, you mix that into the into the solution. Then you can easily track down the DNA sequence information. So for instance, if you have your fragmented DNA sequence like in here, then you put the primer. So primer means that the tiny sequence bonds to the template DNA sequence. So template DNA sequence means the DNA sequence you want to know about the order, right? So this is your sample. Then you put the primers like in here. Then this is the tiny short sequence, but it complementary bounds to that area, right? So bounds to this area. Then you mixed with that these two things with the DNA polymerase, which is make you the synthesis, and you put the nucleotides like an A, G, C, T, right? And also DDGTP means that this is the terminated things, right? So if you mixed with that, then if you go to here, so this is the C, so start from here. So this sequence has the A synthesis and G synthesis and do the terminated, right? And it only has this one sequence on here and terminate on here. And that is represented on somewhere on this end. So this is not really 100% one-to-one matchup with these two, but if you put this DNA sequence into the gel and electroprocess, then it flows from to here, then it's sitting on somewhere in here, right? So and this migration depends on the size of the synthesis, right? So these things is obviously the shortest one. So it migrates faster than others, right? And that is the sitting in somewhere in here. But other situation like, based on this template, the same template, but it has the synthesis further, right? So T, T, C, T, C, and accidentally get the DDGTP, which is the terminator, and that is the terminating somewhere in here. So the last one has this C. So you can basically read out this information, right? And then you can have much further than that. So it has the longer, it actually has the longest one, and it also has the terminate by G. So termination is not always by G. It can be A, T, or G, or C. And because of this termination, it can represent it somewhere in here. So you terminate by A, it appears somewhere in here, and by T somewhere in here, by G somewhere in here, by C somewhere in here, right? So based on this information, then you can easily read out which things you can get the sequence composition from there, right? So you can use this information for the sequencing. Okay, so yeah, right. So you can basically read out the sequence information from bottom to up, and this is the shortest one to longest one. Then you can basically read out the DNA sequence information. So here is the T, A, C, G, A, G, T, A, T, A, T, G, G, C, G. Oh, this is them fun things, right? I mean, why I be, slow in this, because, we're not going to use this anymore. So this is the sort of, I would say this is the dead technology. We are digging up the history. So, you probably see one time in your career, but not going to back in this one. But I mean, historically meaningful. So this is the basics of the Sanger sequencing. So you read the DNA sequence information by the terminator nucleotides and read out the information from to here, right? So this is the technology at the very first time for the Human Genome Project, and this is always named by the chemical sequencing method. So at that time people called it Gilbert way, right? So because the chemicals from the company called Gilbert, right? So Gilbert technology. Okay. Okay, so if you want to read out the Sanger sequencing information, you have to fragment your genome. So if you think about the chromosome position, so this is like, very long linear DNA structure. So there is no way you can read out this long linear sequence information by Sanger sequencing. So what you have to do is you have to chop out the long linear sequence into the fragment. So this process we normally call shotgun, right? I mean, Korea is the gun-free country, so we never thought about the gun before you join the army. So I mean, and even you join the army, you only use the two types of guns. So I mean, it's unlikely to know about the gun, but there's a gun, the type called shotgun. So shotgun is like, the bullets is all the spread out by the mechanisms, right? So if you do the fragment to your genome, that situation you call the shotgun genome sequencing, right? So that basically meaning they chop up the DNA sequence into the small pieces, and this small pieces is of course doesn't have the discrete size. It has the the large range of the DNA sequence. So it could be have like the 1 kilobase pair length, or sometimes 2 kilobase pair or 10 kilobase pairs, right? Because it's the randomly stochastically breakout, right? So, but anyway, it's a breakout into the pieces. Then you use each pieces. Then you do the Sanger sequencing. But for doing the Sanger sequencing, you only have 1 or 2 tiny sequences. So what you have to do is you make amplification of these sequences. You have to basically copy out this sequence multiple times. Then you increase the amount of input into the Sanger sequencing. So that situation, you use the cloning technology, right? So you do the fragmented at the first time, then put it into the vector, then you grow them in the cloning, and after that you do the Sanger sequencing. Then you can read out the genomic information from there, right? So there are many key terminology come out from here, and I mean, I would say this is still that technology we're not going to use anymore in these days. I mean, it's only few companies they are still using it, but I mean, given your career, you're not going to work in that situation. So I wouldn't say this is the the essential technology. You have to know that, but the terminology from this situation is still using in a latest technology. So some of the things you have to understand, like shotgun genomic sequencing, or the fragment genomes, right? Or like clone things, right? And also the primer, right? The primer is always the situation like the small, tiny DNA sequence complementary bounds to the template sequence, right? And also after that, you have to assemble, you have to concat that sequence into the large pieces, the entire DNA sequence. In that case, you call this fragmented area and put it into the Sanger sequencing, the base pairs we call the reads, right? So reads means that you read that region, right? So reads is the fragmented sequence from the Sanger sequencing. So that case we call the reads. And after that, you use these overlapping reads, then you do the assembly. You basically infer the entire sequence out of that. So even though this is the dead technology, you still have some core terminology still using in these days, right? Okay, so time's up. So we are resume our session from 28th, next Monday, Wednesday, no class, finally. So enjoy your holiday this week, but you know, that doesn't mean that you're not going to review your class. So review your class, also leave the questions on Slack channel, and also please be on the mid-semester survey so I can, you know, see how you feel during your class and how to improve your performance in the class. So I mean, ultimately you all want to get a good grades from here, but my dream is also, you know, you all get good knowledge from the class. So I want to feed up the good things to you. So please be on the surveys, then I can come up with some you know, the brainy solution for that. Okay, so it's all good. So everyone good on the exam and class, and see you next Monday.